Okay, so welcome to our chat today with Dr. Bruce Verchere. We're catching up with him. He is the 2023 Lifetime Achievement Award recipient in Diabetes Canada from Diabetes Canada, and he is a professor at the Departments of Surgery and Pathology and Laboratory Medicine at UBC, or otherwise known as University of British Columbia. He's an investigator in the Childhood Diabetes Laboratories at BC Children's Hospital and director of the UBC Center for Molecular Medicines and Therapeutics. He also holds the Irving K. Barber Chair in Diabetes Research and leads the recently created BC Diabetes Research Network. His research focuses on understanding pancreatic islet function with the goal of developing therapeutic approaches for enhancing beta cell survival and function in diabetes and following transplantation. He has published over 150 papers in the field. He was awarded the Diabetes Canada Young Scientist Award in 2006, a Queen Elizabeth II Diamond Jubilee Medal for Diabetes Research and Service in 2012, and the Jerry Grodsky Lecture of the Western Region Islet Study Group for 2022. He served on a number of advisory and editorial boards as chair and member of grant panels for CIHR, NIH, and JDRF, and is currently an associate editor at Diabetes. Thank you so much for joining us. Sorry, just letting someone else in. And feel free, those who are participating, to drop any questions you may have in the chat as we go along. So thank you so much, Bruce. This is really an honor for us to have you talking to us and taking the time out of your busy schedule to do so.
Well, thank you for having me.
Oh, yeah, absolutely. I just want to ask, just a little bit about your personal history in islet biology and T1D research, kind of high level, just sort of walk us through, your approach and maybe sort of, where you are right now.
Sure. Well, my training was in islets from way back in the 80s. I actually did my PhD at UBC in Vancouver on incretins, the gut hormones that regulate islet function and are now common components of obesity medications. I worked on GIP, so that got me interested in islets. I was in a gut hormone lab, and so I stayed in islet biology. I went and did a postdoc in Seattle with Steve Kahn and Dan Port. They were really a type 2 diabetes focused lab, but that's sort of where I cut my teeth, I guess, in islet biology. And then when I came back to Vancouver in 1997 to work at the Children's Hospital in UBC, because I was at a children's hospital, there was interest in childhood diabetes, type 1 diabetes. And so I just started to get drawn into it there from a background of islet biology, not really with a background in immunology, but there were a lot of immunologists there to work with. And I guess at that time, it was starting to become recognized that beta cells play a role in type 1 diabetes. It was okay to be a beta cell biologist and maybe a bit naive in immunology to work in that field. And so I was just drawn into it. There's this great Canadian research history in type 1 with Banting and Best, and was surrounded by really great scientists in the area, and so was drawn in. And also funding, of course, always helps with funders like Diabetes Canada and JDRF to move your research in that direction.
Yeah, well, I mean, sort of as it stands, of the five, would you agree that the four big centers in Canada are UBC, Toronto, Manitoba, and is there one, well, maybe McGill, I'd say.
Yeah, did you say Alberta? Alberta.
Oh, Alberta, University of Alberta with Patrick McDonald. What am I doing?
Yeah, you're going to get me in trouble here because of all my Canadian colleagues, but both University of Alberta, Pat McDonald, James Shapiro, Greg Corbett, that group that really pioneers in islet cell therapy, and University of Calgary with Per Santamaria doing nanomedicine work and immunology, and Toronto and Vancouver, of course, Montreal. So yeah, it's a great community, very collegial community, strong in islet biology, growing in type 1 diabetes.
Yeah, no, it really represents. We've had many scientists from Canada contributing to the podcast and also to our talks to sort of showcase their work, which has been very strong. I wanted to sort of see, these are some big questions, But why do you feel that the field of T1D is sort of like at an exciting inflection point? And why
Mhm.
why would you encourage trainees to enter the field?
Right. Well, it's an exciting time in science in general, right? With new technologies, regenerative medicine and stem cells having a potentially a huge impact in type 1 diabetes with cell therapies, CRISPR technologies, genomic editing, nanomedicines. And big data and bioinformatics and repositories are really sort of changing the way we do science. Things that we sort of thought were impossible or dreamed of years ago, you can do in a few days in your lab now. So it's a really exciting time. And then to apply these to a disease, a complex disease, still a real puzzle, uh, type 1 diabetes. I think there's such great potential. There's a lot of resources at our disposal now, databases. I mentioned these biorepositories like NPOD and HPAP, human islets. You mentioned University of Alberta. So our access to tissue and data has never been better, and that's really changing our approach. I think the way we do things. The path to clinical trial seems faster. You've seen, I mean, it still takes a long time to go from lab to the clinic, but even just in the last few years, the cell therapy and immunotherapy trials, far more than we used to see when I started out. And I think too, for me, one thing that's really changed my approach and inspired me is working with people with lived experience with type 1 diabetes, children and their families at Children's Hospital, really working with them as partners in the research and with the funders as well. Groups like JDRF are much more involved in moving the research forward sort of as an iterative process. So it's a really exciting time scientifically, but the possibility that you could, do some good and help people living with this disease is I think an added thrill and benefit and source of inspiration for me actually working with them.
Yeah, that's well put. And it's interesting, sort of from our, wide interaction with the scientific and medical community at large. There's so many people who actually have the disease who are scientists in the field and
Mhm.
clinicians in the field. So it really is, it's kind of an interesting field.
I think
I,
we all have people, sorry to interrupt, we all have people in our labs working in the labs doing their PhDs, postdocs who live with type 1. There's two in my lab now, and they bring a very unique perspective for us. We try not to always ask them questions, but about their disease, but they're, quite willing to talk about it with us, and they provide this sort of bridge between the patient community and the lab, which I think is really valuable.
yeah, I agree. That's really great. Um, let's sort of shift gears for a minute. I'm going to ask some, more scientifically specific questions after this big question, which is, what do you envision is the roadmap to understanding the etiology of T1D?
Yeah, it's a big question. It's a complex disease. As you know, as we know, it's multiple genes involved, many genes, environmental factors that aren't as well understood as we'd like them to be, probably viruses and other aspects of the environment. We still don't have a great handle. We know there's tens of genes, 50 or more that confer risk, but we don't fully understand how they confer risk. Some are involved in immune function, some are involved in islet function, many are involved in both. And so we need to understand that. Those studies take time. You have to dig in the lab, but you also need big patient populations, particularly for understanding the role of environment. These studies can take years, right, following populations. But we're making progress. I think, the way to get there, roadmap, if you will, is working together, teams, consortia, data sharing, getting groups together, bringing the islet biologists and the immunologists together, the clinicians, the trialists, the bioinformaticians, I think all working together. And you're seeing that more and more. And I include the people living with type 1 diabetes and with lived experience as partners in this as well. But you're getting people in the room together and talking it out and doing team science. We really need to bring in different points of view, different expertise, different technologies.
Yeah, and I would even offer, I think it's great that some scientists are coming, from very interdisciplinary fields that maybe aren't even focused on type 1 to bring their expertise to the question, to
I
question.
think that's so true. If you look at how um, technologies and their application to type 1 diabetes, glucose monitoring and pumps and now looping, that was really getting the engineers, in the room, right, with the islet biologists, with the clinicians and people working on glucose control and people living with the disease. And look where we are with technologies now, right? It's just been incredible sea change over the last decade or so.
Yeah, proof of concept. So if we can have some more communication and collaboration with oncology and perhaps even, scientists who really were deep into COVID.
Yeah,
Uh,
no,
you
this is,
know,
sorry, this is really important. Like you mentioned oncologists, it's not just the technologies, but it's also the ideas from other diseases. One area where we work is in that the regulation of autoimmunity, or what triggers autoimmunity, and sort of the permissive environment for it, and how we could alter that. And you can learn a lot from the immune response to cancer biology, right? About checkpoint inhibitors, but also we're very interested in macrophages and their role. And in tumors, macrophages are sort of helpful for tumor growth. They take on a pro-regenerative sort of anti-inflammatory type of phenotype, it's thought. And so can we harness some of that work from the cancer field to understand what's going on in the islet in type 1 diabetes?
let's kind of just dive into that idea a little bit. So in the islet, right, you've got these macrophages, kind of, the residential macrophages. What if you could do a thought experiment and sort of just or hypothesize, everything's, maybe the pancreata is of someone who's on their way to type 1. Oops, let me just add someone, is maybe a little bit smaller, from the get-go. And maybe they have an HLA component that predisposes them to this. And maybe they got, CVB, they've got a viral insult. And then they, somehow are on antibiotics and their gut microbiome change, all these things, a couple of triggers. And then when you think about the cell biology there in the islet, what do you think is, if you could imagine it, what would it look like? Like alphas, betas, and the macrophage. Do you really think the macrophage is the first initiator of a response to a sickly beta cell?
Yeah. Well, I mean, we know that, that innate immune response is the early response, right, to
Mhm.
in inflammation, to wound or infection. And so it's important. Um, and, studies in the NOD mouse model, and also coming out of human work from NPOD, we know that there's a lot more macrophage. I think we've thought it's a T cell mediated disease for many years, and now recognizing that there's this complex interplay between the islet cells and not just the endocrine cells, but that macrophages play an important role. There, we study them now a fair bit in this context because they're so interesting, right? There's not very many in a healthy and a non-diabetic islet. There's maybe 5 or 10 macrophages in an islet out of a few hundred or thousand cells, but they play really important roles in talking to beta cells, in islet remodeling and regeneration, and then also in the inflammatory response. So whether studies in mice suggest that they're a very important trigger, aminoaminoase work indicates that you can't get type 1 diabetes without that inflammatory response of macrophages. We don't have such a good handle on it in humans, although there is some clinical data, right? The glutamab trial targeted TNF and worked pretty well. And one could argue that the macrophages might be a primary source of that within the islet. So we think they're important. We think they're actually a really good target. In some of our research in the preclinical models, we're targeting them with nanomedicines because phagocytic cells like macrophages and dendritic cells really, they eat up the garbage, right? That's what they do. So they're a fairly good target, really good target of nanomedicines and nanoparticles. So you can deliver drugs to them and then try to, let's say, reduce their inflammatory phenotype or induce them to make regenerative factors in the
If
islet. So,
if you're talking about like, in each in a human islet or a mouse islet, there are five, macrophage and residents. Can you talk a little bit more? Can you extend that into sort of like the M1, M2,
yeah,
you know, axis?
right. So in sort of their basal state, when they're doing their job as residents in the islet, they're thought to be kind of M1-like, or people don't know what that is. That's the inflammatory side, but sort of quietly M1. They're not really inflammatory. And then in response to danger, they can become more pro-inflammatory. And many people, we think too, that this could be an important trigger in type 1 diabetes. Now, what induces them to become pro-inflammatory and take on that phenotype, we don't know. But, viruses could be, for example, one smoking gun there. That's one possibility. But interestingly, one of the things we found in our research is that just in response to beta cells dying, for example, when a macrophage goes to eat up that dead or dying beta cell, they don't tend to become more pro-inflammatory. They actually take on, this M2 type phenotype, which is more pro-regenerative. And so we think that may be an important pathway for what we call islet remodeling, making new beta cells, restoring insulin secretion, which is really interesting, right? Because then it makes you think that if you could somehow move between that M1 and M2 space or phenotype, you could approach that therapeutically, right? So that's one of the things we're doing. If you could sort of make them less inflammatory, less M1-like, and more pro-regenerative, more M2-like.
Yeah. Yeah, I know people are exploring that. What if we talk a little bit about this M1, M2 sort of ballet here in terms of beta cell senescence, which is a big topic right now. Peter Thompson at Manitoba, he came out of Anil Bhushan's lab at UCSF, and they both, are working on this hypothesis of the fact that some beta cells become senescent as they, undergo insult and basically go offline. Has anyone looked at the M1, M2 phenotype in this context?
No, not to my knowledge. Interestingly, we do collaborate with Peter. He's an excellent young scientist, diabetes researcher. We were really fortunate to bring him back to Canada after he trained with Anil in San Francisco. I think the way, beta cells change over the course of type 1 diabetes, early on in the disease, how they respond under the stresses and become stressed themselves, and how they may trigger the immune response, and then how they may undergo these different phenotypes. We already know they're heterogeneous, that there are, there's some debate, but there's, some hub cells or pace setter cells that are small population. There are that have differences in gene expression and function. And then in type 1 diabetes, there seems to be this, and in type 2 for that matter, there seems to be this subpopulation of cells that become senescent. And what we don't really understand is, whether those are sort of cells that are resisting death, they're hiding out from the immune system. One thing we know from human studies now is from NPOD and other work is that type 1 diabetes in humans is not just a disease of beta cell death. There are residual beta cells around that are there. They're potentially a source or a target for regeneration, which is great. But what we don't fully understand is how those cells avoid the immune attack if they're sort of less differentiated or something, or senescent, and somehow they're hiding out from the immune system. So that's a really exciting area of research in its early days. Peter's data from his work with Anil really suggested that, targeting those senescent cells could impact type 1 diabetes, at least in the NOD mouse. So that's, as I said, still early days, but it's a really exciting area.
Yeah, it definitely is. And you just sort of tapped onto some of the work I was going to ask you about, your thoughts on sort of the Guy Rutter, again, another Canadian scientist, and the Richard Benninger work that does indicate there are some pacemaker cells in the islet, pacemaker beta cells. And what are the opportunities sort of there in terms of, maintenance or resurrecting these pacemaker cells as they go offline?
Yeah,
After they go offline.
yeah, it's interesting. It's great to hear you call Guy a Canadian scientist, because I still think of him as a British scientist, but he joined us back in Canada, in Montreal, a couple of years ago now, I guess. So it's great to have him in our islet biology community in Canada, which is a really strong one. Yeah, the pacemaker hub cell idea. I think the data is very strong for it, but what they're really doing, we're just sort of at the beginning of understanding that, and there's still some debate on their existence and role. But I think most of us agree that they're there. I mean, everyone believes that the beta cells are heterogeneous, and there's different populations and subpopulations of beta cells. So what's going on in type 1 diabetes? Do they play a role? I don't think we know yet. I think now there's tools, at least in the preclinical models in the NOD mouse, to target them, right? To delete them, for example. So I think we'll start to get there pretty soon with that. I'm sure groups are working on that. And one could also imagine, if they are this important central cell, right, that's kind of coordinating the islet response, they may be an important target in diabetes as well, right? If they're still around, target for regeneration or improving function.
Mm. Yeah, or keeping them alive in the first place so that they
Yeah.
can sort of run the show. Just, again, if anyone has any questions, please drop them in the chat and we can ask them as we go. I had another question about, well, sort of like one more constituent of, I mean, there's, a number of cell types in the islet, obviously, but I just wanted to ask one more question until I shift gear to the immune side of the equation. But with the alpha cells, is your lab, kind of digging in there a bit recently, or who at UBC is kind of following up on that?
Yeah, there's lots of people working in the alpha spell space now. I think we recognize, from the work of group at Duke, for example, Dave D'Alessio and John Campbell and others, the importance of alpha cells in regulating beta cell function. So they do, alpha cells do more than just produce glucagon as a counter-regulatory hormone to insulin, right? They
Well,
support.
yeah, well, I mean,
Yeah.
they're connected by gap junctions, correct?
Yeah, right. And not just that sort of, intercellular communication, but also through secretion of hormone, probably, and maybe release in the paracrine space in
Fair.
between alpha cells and beta cells, and actually acting on receptors, right? So if you don't have alpha cells around, or they're dysfunctional, that's detrimental to beta cell function. And we know in type 1 diabetes that, alpha cell function, there's dysfunction, right? They don't work as well as they should. The cells are not lost, they're around, but they're dysfunctional. So, are they a target? Yeah, I think they are, potentially for beta cell protection and for therapy, for example. That's hasn't been explored much in type 1 diabetes, but certainly in type 2, there's work in that space. So we're not really, I mean, it's hard to ignore alpha cells, right? If you're working in the islet, we don't study them directly, but particularly in human islets, where the alpha cells, more so than in the mouse, the alpha cells are sort of scattered throughout the islet, and they seem to talk a lot more with beta cells. So there's really interesting work going on in this field. We don't have a lot in Vancouver, but I'd say in Alberta, Pat McDonald's been doing some really interesting work with at the single cell level in human islets with his patch-seq work. And really now with access to human cells and the technology, starting to learn a lot more about what they do.
Yeah, well, that's a really interesting horizon to keep an eye on.
Mhm.
I wanted to, kind of shift gears over to the immune side. Obviously, that's part of the equation. You had a great review in Endocrine Reviews 2021, volume 42, the beta cell in diabetes, integrating biomarkers with functional measures, which was, Stephen Kahn et al. And you.
Mhm.
And so you kind of, talked a bit about the broad application of autoreactive T cells as reliable biomarkers, the deep phenotyping of them. And then, it was a great review, but, there was some indication there because of limitations, the use of autoreactive T cells as biomarkers for type 1 is currently limited clinically. What do you think about that? Can
Yeah,
you
first
expand
that
on
was
that?
really fun review to write because Steve was my postdoctoral mentor from more than 25 years ago. So you're supposed to go on to become an independent scientist, and leave the nest. But it was really fun to go back home and write that with him. It's a really important field, right? Because if we're going to prevent type 1 diabetes, speaking to type 1 now, we need to predict, right?
Mm.
And we're limited there, very much based on autoantibodies for the prevention trials. And so you're asking about other immune cell predictors, and I think it's early days for that. Although, I think where the field is really going is in the genes that confer risk, genetic risk score. I think that's going to really transform prediction. And I think where we work, and I see some colleagues on the call here in the space too, Carmela, others who work on beta cell biomarkers, right? We're believers that the stresses on beta cells induced by the early immune response are detectable with impairments in beta cell function. We're interested in the prohormones, proinsulin and pro-IPP, which seem to be disproportionately elevated early in the disease course and maybe even before. But it needs to improve, right? Because preventing disease would be incredibly valuable. We could do this potentially in a patient-specific way, a precision health manner, right? If you were able to assess from different biomarkers, whether they're genetic risk score or immune biomarkers or beta cell biomarkers, what would be the best approach to prevent in a certain person or population. And
Yeah.
I think that's where we need to go. So that's partly why we wrote that review. It wasn't just focused on type 1. We were very interested in type 2 diabetes as well, and particularly how beta cell function changes over the course of both forms of the disease. But yeah, I think the biomarker field in type 1 is critical for improving disease prediction.
Right. Specifically with the advent of these new medications that are, Tzield and others that are coming, it would be great to kind of get buckets of endotypes
Yes,
and
yeah,
understand who will benefit best for certain medications or combinational therapies.
right. We now have therapies that work, right? Tibulizumab delays disease in people at risk, and some of the work is starting to suggest, like Emily Sims and others, that you may be able to predict from biomarkers who will respond better to these drugs, and now we're getting a much wider set of these therapies, right? Immune therapies. You probably just saw I'll get the name wrong, the baricitinib
Yes,
trial. Just
that's a tongue twister.
yeah. So there's more and more of these therapies. 10 years ago, immune therapy was a bit stalled, right? Frankly, and some skepticism, and people like Kevin Harold and Carla Greenbaum and others were sticking to it. And pushing it forward and being, pretty stubborn about it. And look where we are now, right? It's a long slog with these clinical trials, right? I would say that even the immune therapies now we're using are, it's still a bit of a sledgehammer approach, right? They're general immunosuppressants. So I think where we would love to go in this is in an antigen-specific way, so that you can go in with a magic bullet, so that you're not just doing a general suppression of the immune system and
Yeah,
some of the risks that go along with that, but going in and
yeah,
targeting those immune cells which are autoreactive to beta cells specifically. That,
yeah.
if we just say one more thing too, sorry,
Oh, no.
one you talked about uh, combination therapies and so now there are a couple of trials targeting beta cells too, right? Which is new as well, right? Verapamil for example, to
Mhm.
relieve beta cell stress. And I think where you'll see more and starting to see it for various reasons, there's been challenges in doing this, but to have combination therapy trials that target both the immune system and the beta cell.
Yeah, that would be ideal. Yeah, we had Anath Shalev speak with us, like almost 2 years ago, and that was a fledgling, almost like a fledgling hypothesis, closet hypothesis, and then suddenly it just really burst onto the scene. So that was exciting.
I think yeah, surprised the field a bit, right? For those of us who work in beta cells, we were pleased because we've been trying to make the case that beta cells are important in the pathogenesis. But I think this was, one of the first sort of proofs of principle in humans that you can target the beta cell and can have positive outcomes in type 1.
Yeah, and not unlike Kevin Harold, she kind of had to really um keep that alive, keep her hypothesis alive, keep bringing it up to get traction. So I think the field also would benefit to, I mean, of course, science needs rigor, but to be open to new ideas. And I just wanted to tap into one last thing. Um, you know, the idea of going back into historic data to drive new hypotheses. What's your impression on that? Obviously, it seems as if people are now diving back into Teddy and Daisy and pulling out novel hypotheses with new tools and new ways of interpreting these data that have been sort of like, you know, uh, I don't want to say, well, they've been generated and they're kind of hanging there for the picking. So what are your thoughts around that?
Yeah, I think we're just starting to scratch the surface of that, right? And it speaks to the importance of these repositories of data, right? These big, long-term studies following whether it's screening or following people living with disease, longitudinal studies, and the wealth of data that gets generated, and also the repositories, right? Like HPAP and NPOD, and then how we access that data, right? Because if we're a beta cell biologist or an immunologist, we likely do not have the expertise to really dive into that data in the way we should. And so this really speaks to partnering with the computational biologists and the bioinformaticians who, we can ask the questions maybe, but, don't know how to really optimally access that data and use it. So I think, yeah, there are good answers to important questions hiding in big data and in these repositories that you're going to see. They're starting to trickle out now, but you're going to see more and more in the coming years. It's a
Yeah.
really exciting time.
It's really exciting. And again, it taps back to what you mentioned earlier, the importance of team science.
Yeah, and data sharing and making the data publicly available, which I think these NIH and JDRF, the funders, are making that requirement, making it not just available but accessible, because it should be user friendly, right? And
Totally.
easy to access, right?
Yeah, not always easy. I know, we ran a couple of data-driven challenges for early career scientists, and they're like, how can we get access to Teddy? Sort of a long road and an application, and I think hopefully that will become more, um, oops, here's one more person.
And
Um,
and I
it'll be facilitated, as people understand the power of accessing these data.
yeah. I think it also speaks to the importance of engaging the community of people with lived experience with diabetes too, right? The patients, the families, they really need to participate, right, in these trials and screening. And the more we have, the better the data, right? So I think, and increasingly working with patients as partners, I think has really started to transform our approach for sure. In our JDRF Center in Vancouver, it's not just talking to them about our discoveries and the research we do there. It's a two-way street, learning from them what's important to them, what a cure means to them, right? And where they would like our research to go, and also teaching us how to better communicate the results of our research.
That's a point well taken. And I think there's a lot of work being done there though. TrialNet is working, T1D Exchange reaches out to patients beyond type 1. They're all patient facing and they're trying to bring more people forward so that they can understand, how important it is for them to participate. So I think they're doing a great job. Um, Okay, well, I mean, I don't want to take too much more of your time because it's been great. I have a couple of questions here in the chat. Regarding the M2 polarization of macrophages you mentioned, there's evidence from developmental studies that macrophages facilitate remodeling that is important in duct development in the embryo. If the macs in the diabetes setting are shown to be more M2, do you think they could only be regenerative if the surrounding environment is receptive to the remodeling? That's from Teresa Mastracci, a great
Yeah,
young
no, thanks,
scientist.
Teresa. It's a good question. I will say, so the short answer is I don't know that, but the short answer is yes, for sure. But I think what we're learning is that the phenotype of macrophages, whether they're pro-inflammatory or pro-regenerative or something in between, depends very much on the environment they're in. So what they're seeing. So if you, for example, deplete macrophages, in a model system, and you sort of let them repopulate the islet or another tissue, they're profoundly influenced by what they see. And so, during remodeling, when beta cells are lost, as I said before, I think there's something in the sauce there, something they see, whether it's a signal from the dead and dying cells in the absence of some danger signal that induces them to become more pro-regenerative. I think we have a lot to learn from the developmental side of that, though, because we know that if you deplete or delete macrophages from development, the islets don't form normally. So we know that they're critical in development. Not sure if that answered your questions, but we can talk about it sometime.
She also is very involved in animal models such as mouse and zebrafish. Zebrafish, quite important in that space.
Yeah, the
So,
developmental biologists have really, I think, had profound effect right on the field. We wouldn't be where we are with stem cell, derived insulin producing cells in transplantation without all the work in developmental biology, for example, and how the pancreas normally develops.
yeah, so, out of the Melton lab and et cetera, that they wanted to sort of emulate that, which they have, looking like they're very close. So, I would just say, we also got a nice kudos, congratulations. Bruce, amazing work and so well deserved on the award from Carmela Evans
Well,
Molina.
thank you, Carmela.
Yeah, she's another sort of powerhouse from the Indiana team. They are just doing such great work there.
They are, and they're a great group to collaborate with, and they're a great group to socialize with at meetings, which we do sometimes. And sometimes that's where some of the best conversations happen, right, that lead to further collaboration. So we're working together in this space around residual beta cells and prohormones as biomarkers of beta cell function in type 1, and it's been really fun and rewarding.
Fantastic. Well, we hope to keep an eye on papers coming from your laboratory, and certainly we'll feature them as they come out. And we so appreciate you taking the time right before this busy holiday season or during the busy holiday season to talk with us and sort of talk through some of your ideas and where your thoughts are in the space. Really appreciate it.
Yeah, thank you so much, Monica. I love the sugar science. I think it's great what you're doing, bringing diabetes science to everyone, to not just the public, also other scientists. I love listening to your podcasts and shows, so really appreciate you having me on.
Thank you so much. And we'll be posting this recording on our website shortly. So have a great holiday, and we'll speak to you soon, I hope.
Yeah, thanks. We'll see you at a meeting or talk to you soon, and happy holidays to you too.
Thank you. You too. Bye-bye.